Hello, in this video we're going to talk specifically about proteins. In previous videos we've mentioned or began to mention how proteins come together to produce the quaternary structures that give enzymes the ability to function the way they do. But in this video we're going to kind of dive in and talk about an in-depth discussion of how proteins structure themselves and how those structures impact ultimately the function that that particular protein can produce. Okay, and so if we talk about how proteins are structured, we know that all proteins consist of a single monomer called a peptide or an amino acid. These amino acids, which can be seen over here on this top right, consist of three parts. We're going to get into the actual structure of the amino acid in a minute, but peptides, remember, are individual amino acids. Okay, and these individual amino acids or peptides are linked via peptide bonds, and the way that they are linked is a condensation reaction, just like all other monomers when built together or put together are linked by removing water. We're going to talk about how the ribosome actually does that in a minute, but proteins at their simplest nature, their simplest structure, are long chains of amino acids or peptides that are linked via peptide bonds. We're going to see a diagram that shows a chain of those in a minute. But if you can imagine, these single monomers, these single peptides that are linked in repeated fashion would constitute a protein. And they are produced by a ribosome. Ribosome is a eukaryotic and prokaryotic organelle that is going to locate itself either in the cytoplasm, attached to rough ER, the rough endoplasmic reticulum, or the cytoskeleton. And the ribosomes are going to be the home for, or the site for, the process of protein synthesis. Now obviously protein synthesis is the building of a protein by linking these amino acids together. That happens in the organelle ribosome. Once these ribosomes produce a protein, they are going to be slightly modified by the rough ER, but they are most going to be modified, or they're going to be most modified by the Golgi apparatus, which again is another organelle that is only contained within eukaryotes. Okay, within eukaryotes, Golgi apparatus is kind of analogous to the post office's office. It's going to receive, unpackage, modify, relabel, repackage, and then ship those proteins everywhere within the cell, and sometimes for export out of the cell. Protein folding, once the protein has been completely produced, is going to be initiated and held together by three different things. These hydrogen bonds, salt bridges, and disulfide bonds are three entities that are going to help hold these proteins in the quaternary and tertiary structures that are synonymous with protein structure. And by folding the proteins using these hydrogen bonds, salt bridges, and disulfide bonds, we're going to either produce a fibrous protein or we're going to produce a globular protein. Globular proteins are going to be most often soluble because they are going to be structured in a way that allows the hydrophobic regions to be kind of folded into the center of a quaternary protein, like this one right here on the right. If you put all of the hydrophobic regions deep down inside of this quaternary protein, then it can actually dissolve in a polar substance like water by keeping the hydrophilic regions on the outside. Okay, fibrous proteins are often not soluble, and that's because they are long and stringy. They don't have the ability to put or hide the hydrophobic regions into the center. They stay long and fibrous, and so they are always exposed, and by they I mean the hydrophobic regions. Hydrophobic regions being exposed are not going to allow the fibrous proteins to dissolve in water, but most often they don't need to dissolve in water because they are used as a structural protein, not a functional protein. Okay, these globular proteins are often functional proteins, and so therefore have to be dissolved in solution in order to do the things that they're supposed to do. Okay, if we look at the individual amino acids, you'll notice that there are three components. You have an amino group. The amino group is characterized by the nitrogen, okay, nitrogen being the most important within the amino group. NH2 would be the amino group. We have a carboxyl group, which is a COOH group, okay, carbon, oxygen, hydrogen, and oxygen, COOH group, and then you have a radical group. Radical group is the most important in terms of amino acid variation. Okay, every single amino acid known to man is going to contain the same exact structure except for the R group. Radical group is what produces the variation among amino acids and then ultimately the variation among the proteins. Okay, and so if we look at how the amino acid structures itself, specifically the radical group, notice that in every single one of these examples, and there are a bunch of different varieties, we have kind of classes of amino acids, if you will. We have this nonpolar class. Okay, these amino acids up here are nonpolar. These amino acids are polar. And then these amino acids on the right are either a positive or a negative charge. In every single one of these classes, all of the amino groups are identical and all of the carboxyl groups are identical. In fact, you will not ever find an amino acid that has a different carboxyl or amino group. The only one that is different is the R group. Okay, the R group is what produces the variation and determines which amino acid is which. Okay, and you'll notice that the radical groups, the R groups, are highlighted in every case, okay, because it's outlining the difference between all of these amino acids. Okay, these particular amino acids are nonpolar, these amino acids are polar, these amino acids are negatively charged, and these amino acids are positively charged. And you'll notice that there are 20 different amino acids that are commonly occurring in nature. There are obviously 21 to 22 total amino acids. Some of them are kind of man-made or laboratory-made. We're not going to get into those because this is bio and we're only kind of referring to or focusing on the amino acids that are naturally occurring in nature. Okay, these are the letters, so to speak, that make up all of the words that would ultimately be the proteins. Okay, and so if this amino acid is an A, B, C, D, E, okay, and so on, then we can use or order these amino acids to make different words. Okay, A, C, E is a different word and therefore a different protein than B, A, D. Okay, ordering the amino acids this way is going to produce a specific protein, which we'll say protein number 1. Focusing or orienting the amino acids this way is going to produce a different protein, number 2 in this case. And so protein number 1 and number 2 are going to be made out of different amino acids and so therefore are going to function differently. Okay, these amino acids that we've looked at can be combined in any order, just like I said, and by ordering different letters you're going to produce different words. Okay, and so if we have 20 different options, and some of those proteins could be several thousand amino acids long, you can obviously see that there is an infinite or an almost infinite number of possibilities when we talk about proteins. Okay, amino acids have different properties, polar, nonpolar, charged. We've seen those already within this diagram. Amino acid sequence and individual properties determine how a polypeptide will fold into its protein shape. Remember, most of the time hydrogen bonds between non-adjacent amino acids are going to fold the protein into its secondary, tertiary, and quaternary structures, which we're going to talk about in a minute. But some proteins consist of multiple polypeptides. Some are going to consist of just single polypeptide chains. That's really the difference between a tertiary and a quaternary. Okay, and tertiary protein is going to consist of single polypeptide chains, and quaternary is going to consist of multiple tertiary chains. Okay, quaternary structures are going to be things like enzymes. They're kind of the most complex globular protein that does biological reactions or catalyzes biological reactions. And tertiaries are going to be typically less complex proteins, but a lot of times are going to be more of your fibrous proteins. Okay, how do amino acids combine? I know you've seen several in the last several videos. You've seen several pictures that are showing this exact same thing. This is just a condensation reaction. Remember that individual peptides or amino acids are going to be linked by a condensation reaction. We know that a condensation reaction is going to remove water, and that water is going to basically be made between an O and an H, and in this case another H, but is going to come together to form an H2O molecule. And because we have removed an H and an OH from two separate amino acids, remember this is amino acid number 1, this is amino acid number 2, they are going to form a bond between them and is going to link them together with a peptide bond. Now, peptide bond is just a form or a fancy type of covalent bond, so it is a strong bond. It is not a hydrogen bond, it's a strong bond, it's a type of covalent bond, but the peptide bond is going to hold two peptides together. This is a single peptide. This is a single peptide. Each peptide is held together by a peptide bond and produces a structure called a dipeptide. Okay? Two peptides are held together by a single condensation reaction by removing a single water molecule. This is an anabolic reaction. Remember, we are building up larger molecules from smaller molecules, and two amino acids are linked to form the dipeptide, eventually a polypeptide. So as we continuously link more and more amino acids or more and more peptides onto this growing chain of peptides, we are going to take it from an individual peptide to a dipeptide, ultimately to a polypeptide. Polypeptides are then going to be broken down. Say you're eating, or part of your diet is proteins. Okay, I think protein needs to be a staple within everybody's diet. These polypeptides, once consumed, are going to be broken down using a catabolic reaction, which is the exact opposite of an anabolic reaction, in which we have to put in water in order to split the bond to produce two individual peptides from a polypeptide. How does the ribosome work? So we know that ribosome, which is this green structure in the back, is made up of two separate subunits. This is basically called the large subunit. You can see here. And the bottom is the small subunit. In eukaryotic cells like ours, we have 80 S ribosomes. So I'm going to say eukaryote. In prokaryotic cells, they have 70 S ribosomes. Now, it just refers to the relative size of those ribosomes. Eukaryotic cells have more protein demand, and so therefore have to be kind of a bigger machinery. But 80 S ribosomes in this case, we'll just assume this is human, is going to read, and I don't expect you to know the process of translation yet. This is protein translation. In the process of protein translation, a mRNA, which is this molecule, is going to be read by a ribosome in order to produce the protein that this set of instructions is coding for. And so how is the amino acid sequence put together? It's called translation. We're going to talk about it later on in the IB syllabus, but for now we just need to know the process of linking the amino acids together. We don't necessarily need to know the mechanisms for translation. That's going to be in a later video. We just need to know, like I said, how to link this amino acid to this one, and how to link this one to this one, and this one to this one, and this one to this one. And so those are condensation reactions. We've already outlined what that is and how it works. But ribosomes are read, or ribosomes are going to read the mRNA molecule, like I said, place the correct amino acids in the proper sequence to produce the correct polypeptide. If this message is altered, then the protein that is produced is going to be altered, and therefore the function of that protein is going to be altered as well. Okay, this message that is produced by this mRNA is coded for by a gene. Again, we haven't talked through any of the nucleic acid stuff yet, and so all of that is coming. But for now, the ribosome is going to read a message which is in the form of RNA. It's a type of nucleic acid, and in this case it's mRNA. Messenger RNA is going to be the set of instructions on how to properly produce the correct protein, and it is the ribosome's job to link the correct amino acids in the correct order to produce the correct protein so that the correct function can take place. Okay? If we follow or look at how many possible combinations there are from a potential number of amino acids, we can see very quickly that this number is going to get huge. Every single one of these amino acids, and every single one is an amino acid, amino acid, amino acid, and so on. every place, okay, let's just say this is place number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. If we have a protein here that is 11 amino acids long, and every amino acid location has the potential of having 20 different options, you can see really quickly that the number of different protein combinations is almost infinite. Okay, in fact, the number is so large when we get to extending these proteins to more realistic lengths. Okay, it isn't uncommon to have proteins in excess of 25 to 30,000 amino acids long. Okay, this is the number that is associated with a protein that is 11 amino acids long. You can only imagine how many combinations there would be for a protein that is more realistically lengthened to 25 to 30,000 amino acids long. The number is ginormous. That is the reason why with only 20 amino acids we can produce proteins for every possible situation and every possible scenario. Okay, and the last thing we need to talk about are the levels of protein organization. There are 4 levels. We have primary, secondary, tertiary, and quaternary levels of organization, and each of them are unique, and they just kind of keep getting more complex as you go. Amino acids are, like in the last slide, going to be these repeated monomers that are going to be linked by a condensation reaction to produce peptide bonds, which are going to hold all of the amino acids in a long chain, a long primary single chain of amino acids that are held together by the peptide bonds, which is a form of covalent bond. And the order of the amino acids is going to determine how that protein is eventually going to be folded in subsequent structure levels. So primary structure is a single chain of amino acids from start to finish. Okay? As we start folding those structures into 2 different secondary structures, we're going to get a little more complex. Notice that this is a little different way to look at it. We don't have the cylindrical balls that are representing the amino acids in this case, but you can still see the carbons, you can see the hydrogens, you can see the nitrogens, you can see the CO, which is the carboxyl groups, and so you know that the amino acids are repeated in there. And you also see these dotted lines, which are indicating a hydrogen bond. Remember, a hydrogen bond between this carboxyl group and this hydrogen of this amino group. This is an amino group, this is a carboxyl group, and they are not adjacent amino acids. Okay, this is carboxyl, 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 and then eventually we're at this amino. So this is several amino acids down the line. If this amino acid and this amino acid, which is maybe 5 apart, bonds or forms this hydrogen bond, then it spins this primary structure into this alpha helix structure. So there's 2 different types of secondary protein structures. You have an alpha helix, which can be seen here. It's a corkscrew-like coil structure. And then you have a beta pleated sheet structure, which can be seen here. It's more of an accordion, kind of a folded paper format. Both of these are the interaction between hydrogen bonds between non-adjacent amino acids. Okay, these structures are going to help construct a lot of our fibrous protein structures. Okay, they stay long, fibrous. They are just coiled and folded at this point one time. Okay, those are the secondary structures. So if you take the primary structure and put a coil or a bend into it, and or fold it up into an accordion structure, you have made a secondary protein structure. If we fold it one more time, notice that here we have the alpha helix, which can be seen here, and we also have the alpha or the beta pleated sheets here. You get a tertiary protein, which is a more 3-dimensional globular shape. Okay, we have now taken both secondary structures and we have produced a larger globular protein by utilizing both of the secondary structures in the same protein. This is still a single chain of amino acids, but we have incorporated both of the secondary structures into one protein. It consists of several secondary structures that overlap. Notice that this strand is overlapping these strands, and so we have made a 3-dimensional globular shape. We have started folding based on the disulfide bridges, hydrophilic, hydrophobic interactions, and hydrogen bonding. The hydrophobic regions are going to start orienting themselves in the middle. The hydrophilic regions are going to start orienting themselves on the outside. That way the water that is in solution or water that is in and outside of the cell can interact with this protein the correct way. And the hydrophilic or the hydrophobic regions rather are going to protect themselves and fold in away from the water that is in solution. And this protein structure gives the specific protein the function of actually start working as an enzyme. Okay, you're going to start folding the enzymatic active site. You're going to start folding this protein to allow it to attach to the correct active substrate, and so therefore active site enzyme substrate specificity becomes a thing, and we can then take this protein one step further, join it to another tertiary protein, and you start getting the large very complex quaternary structures that you can see here in a lot of the full enzymes we see, like hemoglobin. Okay, notice that there's a pink, there's a yellow, there's a blue, there's a green. All of those by themselves would be individual tertiary structures, but when we start putting this tertiary together with this tertiary, together with this tertiary, together with this tertiary, we start getting a quaternary protein, which can be seen here. Like I said, a lot of the enzymes that we see throughout biology consist of this quaternary structure. Hemoglobin is an example of that. Hemoglobin is a tetrameric or a 4-unit protein. You can see that there is 1, 2, 3, 4 unit protein. This enzyme is structured perfectly to allow it to bond to oxygen and carbon dioxide. It is housed within the red blood cells of vertebrates like us, and really allows for cellular energetics and cellular respiration to take place by delivering and expelling the oxygen and carbon dioxide. These quaternary structures can produce fibrous and globular proteins, but most of the globular proteins will not become the globular proteins they are without the use of quaternary protein folding. Okay? I think that's where we're going to end it. Those are the levels of protein organization, and this was protein structure. See ya.